Good morning, students. Now in the today's period, we can study here the another bit of our syllabus that is called as DNA replication. आपल्याला हे माहिती आहे की फेडरिक मिश्चर या शास्त्रज्ञानं पस सेगमधून शरीरामध्ये झालेल्या जखमामधून जो पू निघतो त्याच्यामधून केमिकल कॉम्पोजिशन त्याला काही मिळालं, ते ॲसिड लाईक कॅरेक्टरचं होतं म्हणून त्याला न्यूक्लिक ॲसिड असं त्यांनी नाव दिलं. त्याच्यामधूनच आपल्याला DNA आणि RNA हे ॲसिड लाईक स्ट्रक्चर मिळालेले आहे. DNA डबल हेलिक्स आहे. सर J.D. वॅटसन आणि क्रिक यांनी 1953 in the year 1953 gave the DNA double helix model of the DNA structure, and here now in the today's period we are studying here the DNA replication. Replication means the duplication. So we know that DNA controls and regulates regulates the activities of cell. DNA duplicates, and here it described equally in the daughter cell. ते इक्वल प्रपोर्शनमध्ये डॉटर सेलमध्ये जातं, and that process is called as the DNA duplication or the replication. As we know that the genetic informations are carried with the help of DNA, and it performs the two types of function. Now see here, what is DNA duplication? We know that DNA controls and regulates the activity of the cell. We know that this DNA is able to produce the new DNA strand and having equal DNA molecule like the parent cell. So DNA has two types of functions according to its work. Heterolytic function. Hetero means different. Heterolytic function and the autolytic function. Hetero means different. When DNA produces other than itself, DNA सोडून जर काही पार्ट तो तयार करत असेल तर त्याला आपण हेटेरोलिटिक असं म्हणतो. फॉर एक्झाम्पल, प्रोडक्शन ऑफ RNA. वी नो दॅट these are mRNA, rRNA, tRNA. जेव्हा तो DNA सोडून बाकीचे पार्ट तयार करतो, that is called as the RNA. That process we know that it is called as transcription process. RNA DNAच्या वर तयार होतं, त्याला आपण ट्रान्सक्रिप्शन असं म्हणत असतो. See here one strand, and on this strand that is called as template, RNA is produced, that is called as the transcription process, and it produces the another part that is called as the protein also, and that is called as translation. ट्रान्सलेट करणं. भाषेमध्ये जसं आपण मराठी टू इंग्लिश, इंग्लिश टू मराठी, और एनी लँग्वेज वी कॅन ट्रान्सफर, दॅट इज कॉल्ड ॲज ट्रान्सलेशन ऑफ द कोडिंग लँग्वेज इन प्रोटीन सिंथेसिस. सो DNA is performing other functions rather than production of DNA itself. DNA स्वतः सोडून बाकीच्या जर काही बावी गोष्टी तयार करत असेल तर त्याला आपण हेटेरोलायटिक फंक्शन असं म्हणतो. अँड द ॲटोलायटिक फंक्शन, ॲटो मीन्स सेल्फ, ॲटोलायटिक फंक्शन, when the DNA is producing itself the DNA again, that is called as the autolytic function. That process is also called as DNA duplication or replication. Now see here, there are two types of functions, heterolytic function and autolytic function. What is the DNA replication? DNA is able to control and regulate activities of the cell. DNA carries the genetic information and it performs the two types of function. Heterolytic function, it produces the things other than the DNA like the RNA, that is transcription, proteins, translation process. Autolytic when it replicates itself and produces a DNA, it is called as autolytic function. The process is called as DNA duplication. It is semi-conservative type. Semi-conservative. One, suppose these are two strands. They breaks down and produces again here two strands. One is old, another is new one. So one new and one old strand, that type of formation of new strand of DNA is called as the semi-conservative type of replication. This is proposed by Sir J.D. Watson and Crick. They have given that the semi-conservative type of replication. Now see here, there are different stages in the replication or duplication. What are the different stages? Now the activation of nucleotides. Activate करणं. ते activation आपल्याला करत असताना आपल्याला 4 types of nucleotides त्या ठिकाणी न्यूक्लियोप्लाझमच्या मध्ये मिळतात. न्यूक्लियोप्लाझम present, the liquid present inside the nuclear membrane. There are 4 types of nucleotides they are present. They are DAMP, DZMP, then DCMP and DPMP. They are present in the nucleoplasm. They are activated with the help of ATP. It is adenosine triphosphate. That is the biological energy. So ATP work there and it produces DATP, DZTP, DCTP and DTTP. These are activated and this process is called as phosphorylation. So what is the first process? It is called as the phosphorylation. Now here we will find that here 3' end, 5' end and a fork is formed there and 5' end another end is 3' end. The 3' end one end, another end 5' end. Now fork is produced there. That is the replicating fork. So fork is produced there and the leading template and lagging templates are there present. The template which goes from 3' end towards the fork, it is called as the leading template, and the template which forms from the fork toward the 5' end, that is called as lagging template. The RNA primer works there. So here we know that one is leading goes towards the fork and lagging goes away from the fork, which having discontinuous DNA are there. So this is called as the Okazaki segment. We can study later on what are these. Now see here a tuning fork-like structure. Fork-like structure is formed there with the help of both ends, 3' and 5'. The both sides or the templates, they are producing the new type of DNA. So first point is our activation of nucleotide. They activated with the help of ATP, adenosine triphosphate. Now the point or the origin where कोण्या ठिकाणी सुरुवात होते DNA तयार करायला, it is the specific point O and it is flagged by the T site. So it is the O point. It is the unit of DNA when replication occurs. See here, the unit of DNA where replication occurs, that is called as replicon. Replicon is what? It is the unit where DNA replication occurs, and it is a nick point, a specific point. It occurs where it nicks in the sugar phosphate, which is the backbone of phosphodigested bond. So here at a nick or specific point or O side, DNA formation starts here. Now the third, the bonds are broken down and that is the unwinding takes place. DNA is double helical structure. There it breaks down. Unwinding of strands takes place and the DNA molecule gets separated into two halves. That is called as the tuning fork later on. But here the enzyme helicase is used there. Which enzyme is used there? Enzyme helicase is used useful for the unwinding. Unwinding means get separated. The two strands get separated, and at the fourth point, Y-shaped fork is produced. So the Y-shaped or the structure is like this one. Fork is produced, and here the synthesis of new strand also takes place. See here, here pair strand is present, 3' end is there, 5' end is there. Now that is the lagging strand. We know that at the side of lagging strand, discontinuous or Okazaki segments they are present. In the another strand, that is the leading strand, which grows from the 3' end toward the tuning fork, and which go away from the fork, it is the lagging template, that is the 5' end. It always starts from the 3' end, go toward the fork. And now we know that here the synthesis of new strand takes place. One enzyme is used there, that is called as DNA polymerase enzyme. All strands are complementary. Complementary means when there is nucleus base, that is when adenine is there, it's pairing with the thymine, and thymine pairs with the adenine with 2 weak hydrogen bond. When guanine pairs with the cytosine, or cytosine pairs with the guanine, there are 3 weak hydrogen bonds are there. This is the paired strand. This is the paired strand, and we know that here the Okazaki segments they are present, and complementary strands are produced. Complementary means whenever there is at one side A, another side T. When there is Z, another is C. So these are marrying, these are pairing each other. These are anti-parallel and complementary to each other. And now here we know that the complementary strands are produced, and new strand is formed from 5' end toward the 3' end. So the strands they are always forming. Now the leading and lagging strand. What is the leading strand? The template strand with 3' end is leading strand. 3' end is leading strand, and 5' end, that is the lagging strand. Now the replication starts at C3 and ends toward the C5. Complementary strand there 5. And we know that here the tuning fork like structure is present, and that tuning fork like structure which is producing the continuous leading strand, discontinuous lagging strand. On the discontinuous strand, it goes away from the fork, and the segments are present here. These are called as the Okazaki segment. These are called as Okazaki segments. They are joined by DNA ligase. Which enzyme is used there? DNA ligase enzyme is used there. Now after completion or formation of the strand, the RNA primer is removed with the help of DNA polymerase enzyme first in the prokaryote. With the help of DNA polymerase enzyme first, the RNA primer is removed in prokaryote, and DNA polymerase enzyme alpha, or it is used in the eukaryotes. Now the DNA gyrase, these are the names of the enzyme. DNA gyrase is useful for forming double helix of DNA, which forms the daughter DNA molecule. So we have studied the certain steps in the replication or duplication process. These are the activation of nucleotides, then point or the origin, then unwinding, again Y-shaped tuning fork is formed, then synthesis of new strand, leading and lagging strand, as well as the separation of RNA primer with the help of DNA polymerase enzyme first, and polymerase enzyme alpha it is separating, so and formation of the new strand. So one strand is old, one strand is new. This is called as semi-conservative type of replication. It is the semi-conservative type of replication. Thank you.